Hello 7th standard, welcome back. This is our fourth video where we are going to discuss about human digestive system, which is continuation of our third video class. As I said earlier, you have already learned, but still we are just going to discuss what is digestive system and the structure of alimentary canal. Alimentary canal is also known as digestive tract, and digestive system as such include alimentary canal or digestive tract and the associated glands. That we will start. Okay. Now, first, what are you observing in the picture? There are two idlis with the side dish, and then some fish for non-veg people, and then this is a dal what is supplied to you. You are asked to have this as a breakfast, right? And this is sprouted chana. This is green dal. Pacifier this is sprouted. We have done the activity in 6th standard, is it not? Sprouting the moong dal. The same, this is green gram. And these are some fruits. When you are asked to eat these kinds of foods, what are you guessing the components as? Components of food are carbohydrate, protein, fat, vitamins and minerals. Right? So all these food items, when it is taken, how it is going to be digested? That is our digestive system. Right? Now, there are some animals which take different kinds of food and their mode of feeding is also different. What do we know? A snail will just move slowly and scrapping. The kind of food only it will eat some green grasses and the soil only when it is moving on the ground. Mode of feeding only somewhat scrapping. Okay. And then ant வந்து scrapping and brewing. Eagle, it is capturing. Hummingbird, it is sucking nectar. Lice வந்து it is scratching. Scrap பண்றது இல்லையா அந்த மாதிரி. The lice na pain, தலையில உள்ள pain. Mosquito, it sucks blood. Butterfly, it sucks nectar from the flowers. Same way, housefly, it is just chewing and scrapping the food from the items that it is taken in. The table you should know to fill up. That when we discuss the question answer, I will come back to this table 2.1 again. Right? Then this one, first definition for digestion. The process of food eaten taken by it. The process by which the body breaks down the food that is eaten into soluble nutrients. We are taken food as a soluble nutrients. What are the nutrients? As carbohydrate, protein, fat, vitamins and minerals that can be absorbed easily, and this is called digestion. I repeat, the process by which the body breaks down the food that is eaten into soluble nutrients that can be absorbed easily is called digestion. And the steps involved in digestion are first ingestion, digestion, absorption, assimilation and egestion. I repeat, ingestion, digestion, absorption, assimilation and egestion. These are the steps involved in the process of digestion. Right? Now this one has the digestive system or the alimentary canal full structure. Now this as such we name it as digestive system because we are talking about the associated glands also. starting from the mouth, the digestive system with the buccal cavity as salivary gland and the tongue. Continuation is the esophagus and then the stomach and then small intestine, large intestine. This is. And associated glands are salivary gland. These three, one, two, three. These three salivary gland, and then liver is another salivary gland, and pancreas. What I am showing with the pointer pancreas is another salivary gland. We will study the action one by one. Already we are discussing in the previous video class. Okay. So now the mouth consists of or formed of first buccal cavity. Is a full buccal cavity. Now this buccal cavity in human, it is provided with the 32 teeth. Idella teeth on the, this is upper jaw and this is lower jaw. Now the tongue, the pointed what it is showing is the tongue where the one end interior end it is attached and this end towards the teeth they are free. And then at the below the tongue, it is a flap called frenulum. At the cartridge and the frenulum that is getting the tongue is attached with the floor of the lower jaw. Okay, this is the buccal cavity. Now what you are seeing this hanging that is called epiglottis, and this is helpful when the food is swallowed into the space between the tongue and the epiglottis. When the food is swallowed, the windpipe, the pharynx will allow only the food to enter through the food pipe, that is esophagus. Now this teeth in human, there are two types of teeth as it's called diffeodont. That is the teeth which is at the time of birth till certain 2 to 3 years of age, it is called milk teeth. And those milk teeth, they are 20 in number and they are replaced by the permanent teeth. The permanent teeth, it is heterodont. Heterodont, there are 4 different types of teeth as incisors, canine, premolar and molar. Pointed for it. Incisors, in the 4 incisors. 1, 2, 3, 4. In the 4 incisors. இது ரெண்டும் canine. இது premolar. இது ரெண்டும் premolar and they are molars. These 3 are molars. So totally there are 32 teeth are present in human. Now the incisor size of it has a pointed edge. Flat pointed edge so that you can bite the food. So it is for biting. The next canine teeth shape, it is for biting and tearing the flesh. Canine is mainly helpful in carnivorous food. It is for tearing the flesh. And then premolar, it is chewing and mastication. Chewing and mastication. Mastication, the food when it is chewed well in the buccal cavity, it mixes with the saliva and then it forms a bolus. That's mastication. And then it is premolar. Molars. Food chavachu சாப்பிடுறதுக்கு these are all. So the small macromolecule are broken down into micromolecule with the help of this 4 types of teeth as incisors, canine, premolars and molars. These 4 types. Right? Then this one next, it is again the type of teeth in each jaw. First incisors. Incisors பாரு. Upper jaw 4, lower jaw 4. and then its main function is biting and making the food as small slices. Then canine, two in the upper jaw, two in the lower jaw. It is mainly tearing the food. Canine is meant for tearing the food வந்து இங்கே flesh. Carnivorous it tears the flesh. Then premolars, they are four in number and it is helpful in grinding the food. And molars, six in number, further grinding of the food and reduces to a pulp. And the pulp தான் நம்ம வந்து bolus-னு சொல்லுவோம். சரியா? Then this is about the function and number of teeth in the heterodon condition. Okay. Now in the buccal cavity, we have been talking about tongue, and tongue is formed of taste buds as sweet, bitter and sour, and the taste buds are present on the surface of the tongue as the sweet in the tip of the tongue. நம்ம நாக்குல சாப்பாடு வச்ச உடனே ஸ்வீட்டா இருந்தா அது தெரியும்ல. And then at the side sour, sourனா புளிப்பு. And then உள்ள நல்லா கடிச்சாட்டுனா அந்த bitterness தெரியும். அது வந்து உள்ள நாக்குக்குள்ள அந்த taste buds are present. Right? And there are three pairs of salivary gland as parotid. Three pairs of salivary gland as parotid gland, submaxillary or submandibular gland and sublingual gland. Parotid gland, submaxillary or submandibular gland and sublingual gland. சரியா? And these three pairs, parotid, it is present in the cheek. It is given below the ear. அதான் cheek இருக்கு. இந்த cheek இருக்குல்ல, அதான் cheek. Submaxillary or submandibular at the angle of the jaw. இந்த இடத்துல இருக்கு. Angle of the jaw, submaxillary, இது angle. இது வந்து cheek portion, figureல பாரு, pointer. இது வந்து cheek portion, parotid gland. இது வந்து angle of the jaw. It is submaxillary or submandibular, and beneath the tongue is the sublingual gland. So three pairs of salivary gland. What is the function? It secretes saliva. And the content of the saliva are ptyalin. It is an amylase enzyme acting upon starch. So the food which contain carbohydrate will start digesting in the mouth itself. And also it is secreting bicarbonate, salts and mucus. And then there is another enzyme called lysozyme which is acting upon antibacterial agents. It is acting as antibacterial agent. So if any bacteria, harmful bacteria entering along the food in the mouth itself, the lysozyme is acting upon. So lysozyme is also another secretion, normal enzyme சொல்ல மாட்டோம், another secretion which is acting upon the food to kill the harmful microorganism, that is antibacterial agent. And saliva helps to digest carbohydrates in our mouth. So this is for the salivary gland. So you should know the three pairs of salivary gland and the contents of saliva. இதெல்லாம் ptyalin, bicarbonate, lysozyme, and it is acting upon carbohydrate. Right? So once the food is chewed well from the buccal cavity, it is reaching the stomach through the food pipe or esophagus. So when it is moving through the pharynx, it enters the food. It is called bolus. It is entering into the food pipe. That food pipe is esophagus. And when நம்ம முழுங்குறோம் இல்லையா சாலம், that is swallowing. That swallowing, it is the act of swallowing is called peristalsis. The phenomenon is called peristalsis, which is a series of involuntary wave-like muscle contraction which move the food along with the digestive tract. இதை இப்படி சொல்லுவோம் இல்லையா, இது ஃபுல்லா இசபேகஸ் தான், ஃபுட் பைப்பு. ஆனால் இந்த ஃபுட்டு இங்கேருந்து இங்கே வந்திருக்கு. அப்போ இது வந்து பெருசாகிக்குது, இங்கே சுருங்கிட்டு இங்கே பெருசாகிடுச்சு. மறுபடியும் இங்கேருந்து இப்படி முழுங்குவோம் பாரு, அதுதான் பெரிஸ்டால்டிக் மூவ்மெண்ட். இந்த ஃபுட் சூடு ஃபுட்டுக்கு பேரு போலஸ். அதனாலதான் இது வந்து போலஸ் ஆஃப் ஃபுட். ஸோ அந்த ஃபுட்டு வர 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 the muscles will get relaxed so that it is reaching the stomach. So the peristalsis, the rhythmic movements of the esophagus are called peristaltic movement, and the process by which food moves down is called peristalsis. Then this one is about the liver. Liver ku munadi stomach na vandana solli. Now this stomach, it is as lined by gastric layer. Stomach figure na tharla, the figure irukadnala just listen. Stomach is lined by a gastric layer. It secretes the gastric juice, the enzyme gastric juice. And this stomach layer, it is also having another special type of cells called oxyntic cells. This oxyntic cells secrete HCl. So the content of gastric juice is HCl, renin and pepsin. Now HCl is making the medium acidic, making the bolus acidic, and also it prevents the erosion or corrosion of the stomach layer and preventing ulcer. The burning layer of stomach is called ulcer, causing ulcer. And then this renin, it is present in, it is a milk protein present in infants. Infants na chinna kuanda, poranda kuanda. So that infants, when the food is digested, adhu chinna thai paalo illana the powder paal thane kudikum. So adu kudicha it is kakkaradhu, the digestion முடிஞ்சோனே அதை கக்கும்ல, அது தயிர் மாதிரி வரும். அதுதான் digested milk protein. So it is secreting the infant's milk protein. It is acting upon the protein content, and where it is forming a casein. That's casein nu solluvom. Okay. And then peptone, content of gastric juice, HCl, renin and pepsin. Now this pepsin is acting upon protein and it is converting into peptones. That is protein conversion. This is for the functioning of stomach. Now at this juncture, the semi-digested food in the stomach, it is called chyme, CHYME. It is known as chyme. Now when this chyme, when it is reaching the duodenum, next part of the digestive system, as small intestine, it is receiving the secretion of pancreatic enzyme and also liver. Now liver is another digestive gland, but it is the largest digestive gland in our body, and it is reddish brown in color, and it is divided into two main lobes, right lobe and left lobe. Now this right lobe is larger than the left lobe. And on the undersurface, the male part of the liver, இது வந்து கீழே உள்ள the bottom portion of the liver where gallbladder is present. Gallbladder is a structure which is storing bile temporarily. So now this liver, it secretes bile and it is stored in the gallbladder temporarily, and then it is reaching the duodenum for further digestion. I hope you understand. Same way, this content of the bile juice, நம்ம இது படிச்சோம்ல pancreatic, அதே மாதிரி content of the bile juice, it has bile salts as sodium glycolate and sodium tauroglycolate, and bile pigments, bilirubin and biliverdin. And bile salt, they mainly helpful in digestion of fat by bringing emulsification, conversion of large fat droplets into smaller ones. I hope you understand. Now this is the structure of liver. From the secretion while it is reaching the duodenum. Right? Next one is functions of liver. சொன்னேன்ல bile juice, sodium glycolate, sodium tauroglycolate, it is bile salt, bilirubin and biliverdin, it is bile pigments. Functions, it controls blood sugar and amino acid. It synthesizes fetal RBC. Fetal RBC na normal RBCs, it is red blood corpuscle. They are produced in the bone marrow and they are destroyed in the spleen and liver. So, அந்த இதில் தான் the fetal the developing embryo in the mother's womb is called fetus. So, that fetus or RBC synthesizes by the liver, and it produces fibrinogen and prothrombin. These two are plasma protein present in the blood and it is used for clotting of blood. And normally RBCs, they are destroyed in the liver and it stores iron, copper, vitamins A and D, and it produces heparin, an anticoagulant. Heparin is a substance present in the blood actually. It is produced by the liver and it is acting as an anticoagulant. Blood வந்து clot ஆகவே கூடாது, அதான் coagulation. So the presence of heparin is a must for every part of the blood. Then it excretes toxic and metallic poisons and it detoxifies substances including alcohol and drug. So these are the functions of liver. Yeah. Next one is about the pancreas. Pancreas, it is acting both as exocrine and endocrine gland. என்ன சொல்லுவோம்? The digestive part of the pancreas, it is called as exocrine pancreas, and the hormone part of the pancreas is known as endocrine pancreas. Now here the exocrine gland, it secretes trypsin, chymotrypsin, carboxypeptidase, amylase and lipase. What does it mean? The trypsin and chymotrypsin, they are acting upon proteins, and then carboxypeptidase, they are further into peptides and peptone, and amylase, it is pancreatic amylase. If any starch is not digested, that will be done here, and lipase, it is making fatty acids and glycerol. So this is for the exocrine part of the pancreas. Then from there, food is entering into that chyme is entering into the, from the duodenum, it enters into the jejunum, that internal small intestine portion. They are as sucrase, maltase, lactase and lipase. So jejunum consists of 2/5 of small intestine. It starts from the duodenum and ends with the ileum. The secretion of small intestine is intestinal juice, and this is called succus entericus, and that contain sucrase, maltase and lactase. I hope you understand this. So this is for the secretion of small intestine, and then after that the food is absorbed by the villi, the small intestine. It is formed of ileum. Villi is called a finger-like projection like this. This is a single villi enlarged. There is a small projections on the small intestine. They are nothing but blood capillaries. Now these lacteal, it's called a blood capillaries along with the lacteal. Lacteal is a structure where it is absorbing the lymph. It is called lymph. So that small intestine, it is formed of numerous villi which is increasing the absorptive surface area. So that villi, it absorbs protein as amino acid, glucose and other minerals and vitamins, and fatty substances. It is absorbed by the lactase as lymph. Sorry. So all these, and then from the undigested food is passed on through the ascending colon, transverse colon and descending colon and reaching the rectum, and undigested food material is sent through the anus. Thus, in human digestive system is formed of steps of ingestion, digestion, absorption, assimilation and egestion. So ingestion வந்து buccal cavity la, digestion வந்து from the stomach, then liver and pancreas into duodenum and then small intestine. Adala food is absorbed by the small intestine. The digested food is absorbed by the small intestine. The complete digestion of food is taking place only in the small intestine, and then they are absorbed by the villi. Villi are the large finger-like structures present in the small intestine, which is increasing the absorptive surface area. Then once the food is absorbed, it has to be assimilated, synthesized, and wherever the food is needed to the site, it is taken by the bloodstream, and then undigested food materials are sent through the anus. Correct? Now the figure what you are seeing is a cow. Yeah, it is a ruminant animal. What does it mean? Ruminants are animals which are capable of digesting the food with the fibers. Carbohydrates are digested well. Now here this is a digestive system where the ruminants, the stomach, the characteristic feature of ruminant, they have 4 compartments in their stomach. 4 compartments in their stomach as rumen, reticulum, omasum and abomasum. I repeat, rumen, reticulum, omasum and abomasum. In that, rumen is the largest part and the main digestive center. Normally, the cow, goat எல்லாம் நல்ல food சாப்பிட்டுட்டு ஒரு ஓரமா உட்கார்ந்து அசை போடும் இல்லையா? That is called chewing the cud. அதாவது வேற ஃபுட் வந்து கிராஸ் சாப்பிட்டுட்டு இது அப்படியே இங்க வந்து the rumen, large digest stomach cavity, rumen, அதுல வந்து ஃபுட் இருக்கும். அப்புறம் மறுபடியும் அதை வாய்க்கு கொண்டு வந்து நல்லா chew பண்ணிட்டு then it is taken. சரியா? So as rumen, reticulum, omasum and abomasum. And then it has a special type of enzyme to digest the cellulose. Cellulase is a special enzyme which is digesting the food in ruminants. That is the characteristic feature. Whereas this cellulose is not digested easily by the human. It is not having any special enzyme like that of ruminants. Right? Now this is amoeba, nutrition in amoeba, how it is taking the food. We know that amoeba is a unicellular organism where all the activities are taking place only by the entire structure. As see here, through the pseudopodia, food is entering into that. So it is forming the engulfing the food that is taken, that is ingestion, and then it is entering into the body and forming a food vacuole. And in this food vacuole, food is digested, and then undigested food material, they are sent out as digested vacuole. I hope you understand. இது வந்து ரொம்ப simple. In the figure பாரு. Food, the pink color, food particle through the pseudopodia engulfing. And then full and the engulf பண்ணி, இந்த full engulf ஆகி pseudopodia covering the food. இப்போ வந்து இந்த food உள்ள போயிட்டு, amoeba bodyக்கு உள்ள போய், it is forming a circular food vacuole. And then it is getting digested. So here nutrition in amoeba is by putting forth pseudopodia and then it is engulfing the food particle. I hope you understand. So the formation of food vacuole, digested and then undigested food material, they are sent out as digested vacuole. So this is our book diagram. இது வந்து stepwise clearer பண்ணுது. Pink தான் food vacuole. First உள்ள போறது. So here the pseudopodia, they are forming the food vacuole. அது உள்ள இழுக்கிறது. இப்போ வந்து pseudopodia is covered, forming the food vacuole, and then it is digested. I hope you are able to follow this. So in this presentation, discussion, we have done process of digestion in human with the alimentary canal details, and then about the process of digestion in the stomach, liver, pancreas secretion, then in the small intestine, and then through the colon, rectum, undigested food material is sent out through the anus. I hope you understand this. So here the 5 steps in digestion are ingestion, digestion, absorption, assimilation, and egestion. And we have learnt about what? As ruminants, digestion in ruminants with the 4 chambers of the stomach. And then about the nutrition in amoeba, steps involved in nutrition in amoeba. I hope you understand. So in the earlier, in video 3, we have been learning about description of alimentary canal with the secretion. Now this presentation, description plus process of digestion, we have been discussing. I hope you have understood. Yes, sir. Right. Okay. So we'll continue in the next video class. Thank you.